IB Bio Topic 11, Animal Physiology 11.1, Antibody Production and Vaccination. A. This movie has its focus on the specific defense against disease mechanisms, particularly antibody production. That said, there are other details here that ensure full coverage of the IB syllabus within this topic. The essential idea is immunity is based on recognition of self and destruction of foreign material. I've chosen this slide to emphasize an important point within this unit. Every organism has unique molecules on the surface of its cells, as can be seen in this image of a cell membrane. Molecules projecting from cell membranes are unique to each species and each individual within a species. These molecules allow our body to distinguish self from non-self. These species-specific molecules are critical to the defense against specific pathogens. Here is an outline of all the movies available in Topic 11, an HL topic. Higher level IB Bio students use this outline to find the movie you need for review. This movie is focused here. This movie will pick up where Defense Against Disease Topic 6 left off. Topic 6, Defense Against Disease, covered non-specific defense mechanisms, including the skin and mucous membranes as physical barriers to the entry of pathogens. As well from Topic 6, you should remember that phagocytic white blood cells simply ingest pathogens as non-specific defense mechanisms. Also from Topic 6, you should remember the production of antibodies by B cell lymphocytes. Antibodies are proteins synthesized and released by B cells in response to a specific pathogen. While I won't review skin, mucous membranes, or blood clotting as non-specific defense mechanisms, this movie will start with the inflammatory response as a non-specific defense mechanism. Topic 11 includes a brief look at the inflammatory response, specifically allergic responses to allergens. Stay tuned. The inflammatory response is typically initiated by an open wound where white cells of the connective tissue known as mast cells release histamine. Histamine diffuses to local tissues and causes a series of changes in the region around the wound. In this image, notice the capillary is here and phagocytic white blood cells have left the capillary and entered the region of the wound. Histamines, released by white cells known as mast cells, cause the capillary to swell, bringing more blood to the wound area, and blood fluid, plasma, leaves the capillary to cause the area to become swollen. In this image, here on the right, you can see the swollen capillary and phagocytic white blood cells leaving the capillary to engulf infecting bacteria. The inflammatory response is characterized by redness and swollen tissues. Capillaries that have become enlarged due to histamine cause a red coloration to the infected area. As well, the fluid leaving the capillary causes the region to be swollen. These are classic inflammatory response symptoms. An allergen is a substance that causes the allergic reaction. An allergic response to an allergen is, in truth, just a strong inflammatory response. An allergen is any substance that triggers the allergic response. Pollen, mold, dust are examples of allergens. And the allergic response to the allergen is a runny nose, swelling. This comes as a result of the histamine that causes blood vessels to swell, along with blood plasma leaving the vessels to cause the swelling. Allergens, seen here, bind to antibodies attached to the cell membrane of mast cells. Mast cells then release histamine, and histamine triggers the allergic response, a hyperinflammatory response to the allergen. Remember, every organism has unique molecules on the surfaces of its cells. Allergens stimulate antibodies called immunoglobulin E. These antibodies activate mast cells to release histamine. Allergic symptoms include sneezing, itchy runny nose, watery eyes, wheezing, chest tightness, shortness of breath and a cough, raised itchy red rash, swollen lips, tongue, or eyes. Here are the relevant IB syllabus statements that I've just covered. State that white cells release histamine in response to infection and to allergens. Describe the allergic symptoms caused by histamine. Now that brings us to the end of the details around the inflammatory response and allergens required of topic 11.1. But before moving to specific defense mechanisms, let's consider how pathogens, which are typically species-specific, can cross the species-specific barrier. A pathogen is a disease-producing organism, and we do have specific defense mechanisms, specialized cells, B and T cells, that protect us against very specific pathogens. Immunity is the capacity to resist pathogens over time because of memory cells that are produced through an earlier exposure to the pathogen. These cells have remained alive and waiting in the body for a second exposure. Now, before moving forward, I want to say that pathogens are typically species-specific, although pathogens can cross the species barriers. This is known as zoonosis. All of the pathogens on this slide are species-specific pathogens, pathogens specific to the human species. All of the pathogens on this slide are species-specific pathogens, pathogens specific to the human species. There are some pathogens, such as bird flu, H5N1, that can jump from one host to another. Most emerging infectious diseases in humans originate from animal reservoirs from species other than human. There are 4 steps for a pathogen to cross the species barrier. First, the level of human exposure to pathogens must be high. For example, in agriculture, people are in regular contact with the birds they raise. If the birds are host to a virus, the virus could pass to humans. Secondly, changes to surface proteins in the animal or human might facilitate the crossing over of the pathogen to humans. The third step for a pathogen to cross the species barrier is that the pathogen must be able to take over the new host cell's machinery to cause infection. Again, molecules on the surfaces of membranes must have been altered, allowing the pathogen to successfully access the cells of a new host. And lastly, the pathogen must be able to be transmitted among humans, causing outbreaks or epidemics. H5N1 is a highly pathogenic Asian strain that emerged as a pandemic in birds in 2003-04. It has been commonly called bird flu and has resulted in the culling of hundreds of millions of birds in an attempt to control its spread. There is no evidence that H5N1 can be spread by human-to-human contact, but it's possible adaptation poses a potential human pandemic threat for which vaccines are being prepared. Swine flu, H1N1, is also a virus that has the potential to cross the species barrier. Here is the IB syllabus statement that I've been targeting. Using named examples, state that pathogens can be species-specific, although others can cross species barriers. Molecules on the surface of pathogens identify the pathogen as foreign material. The molecules on the surface of pathogens are known as antigens, and antigens activate the immune system of mammals. An antigen is any molecule recognized as foreign that stimulates antibody production. This should be review from topic 6. Memorize this definition of an antigen. In this image, you can see the antigen. This is foreign material identified as such due to surface molecules, mostly proteins, but glycoproteins as well. You can see antibodies, Y-shaped blood proteins produced by the infected organism to defend itself against the foreign material. Antibodies neutralize the pathogen. More on this later. Remember that B cells produce the specific antibody in response to a non-self antigen. So let's review the specific defense system that I covered in topic 6. B cells defend the body in the blood, out in the open, with the production of antibodies against the antigens. T cells have more roles, including the activation of B cells. This is what you need to know. T cells serve to activate B cells. But mostly T cells scan the body for infected cells, cells that are infected with a virus. This is called cell-mediated defense. Here's a great slide to study, and maybe you already see what's happening. The pathogen arrives with its antigens on the surface, molecules on the surface. Every organism has unique molecules on the surface of its cells, and this allows an organism to recognize self from foreign material. The arrival of a pathogen with its surface antigens is the challenge to our body's homeostasis. Our bodies respond to the challenge with cells specifically ready to defend: macrophages, B lymphocytes, and T lymphocytes. Macrophages are large white blood cells that ingest the pathogen, then activate T lymphocytes directly and through the release of a hormone. The pathogen also activates B lymphocytes directly, direct contact. Helper T lymphocytes also activate B lymphocytes. All of the activated lymphocytes are specific to the pathogen. Then the activated B and T cells divide by mitosis to form a clone, an army of cells that will work to remove the pathogen from the body. Notice that the B cells produce antibodies specific to the pathogen. The antibodies bind to the antigens on the pathogen, neutralizing the pathogen. Here is the relevant IB syllabus statement. Outline the principle of challenge and response, clonal selection, and memory cells as the basis of immunity. So let's look at the response by B lymphocytes, B cells for short. An antigen challenge and directly contacts B lymphocytes. B cells are activated. Clonal selection. An antigen, by binding to specific receptors on the membrane surface of B cells, selectively activates a tiny fraction of B cells from the body's diverse pool of lymphocytes. The B cells divide mitotically to give rise to thousands of cells, clones of the specific B cell that was activated. The B lymphocytes produce antibodies that are in the bloodstream and serve to remove the pathogen. Also, some of the cloned B cells are memory cells, cells that remain alive in the body in case of a second exposure to the same pathogen. In this diagram, the antigen activates specific B cells directly. Molecules on the surface of the B cell have a shape specific to the antigen. The antigen is then engulfed, ingested, and the B lymphocyte produces antibodies that, once released, will remove the antigen, the pathogen, from the body. Antibodies are proteins released through exocytosis into the bloodstream, where they will contact the pathogen with its surface antigens. Here is a micrograph of a B lymphocyte with extensive ER within the cell. Remember the process by which proteins are synthesized. A cell, such as a B cell, that secretes numerous protein antibodies would be expected to have lots and lots of ER. See me in class if you've not made the connection to topics that we've studied earlier in this course. Now let's look at the activation of T lymphocytes, specifically helper T cells, by macrophages. The macrophages engulf the pathogen with its surface antigens, then contact the specific helper T cells, cells specific to the pathogen. The helper T cells then stimulate B lymphocytes that are also specific to the pathogen. Let's take a closer look. So here is a macrophage that is engulfing an antigen. Remember that an antigen is any small part of a pathogen that would stimulate the response of these cells. The macrophage digests the antigen and then displays part of the antigen on its membrane as the mechanism to identify and activate the specific helper T cells that will work to remove the pathogen from the body. Now, the molecules on the surfaces of cells are critical to the recognition of self and non-self. The MHC molecules provide recognition of self, while the presented antigen is foreign material to which these immune cells must respond. The activated helper T cells contact B lymphocytes directly to activate the specific B cells that will produce antibodies against the antigens on the surface of the pathogen. Here is the helper T cell activating the specific B cell. The B cell is stimulated to produce and release antibodies. The antibodies will bind to the antigen to neutralize the antigen or facilitate removal of the antigen. More on this later. Here is the IB syllabus statement that I've just explained. You may want to go over the last 4 or 5 slides to review this statement. Explain antibody production, antigen presentation by macrophages, activation of helper T cells leading to activation of B cells, which divide to form clones or antibody-secreting plasma cells, and memory cells. In review, pathogens with their surface antigens stimulate macrophages and B cells and some T cells directly. In turn, macrophages activate specific helper T lymphocytes that serve to activate B lymphocytes. A clone of B cells are formed through mitosis. Antibodies that neutralize the pathogen are produced and secreted into the blood by B lymphocytes. Now, some B lymphocytes remain alive in the body as memory cells, ready to respond in case of a second exposure to the pathogen. This is the immunity we get from first infections or from vaccinations. Here it is in words. An antigen challenges and the lymphocytes respond. An antigen, by binding to specific membrane molecules, receptors, selectively activates a tiny specific fraction of lymphocytes from the body's diverse pool of lymphocytes. This gives rise to thousands of cells, a clone. These are plasma cells that secrete antibodies, and memory cells that provide immunity. Memory cells remain alive in the body over time, ready to respond to the pathogen upon a second exposure. This is immunity. In this graph, we can see antibody concentration in the blood over time following 2 encounters with an antigen, encounter 1 and encounter 2, and the graph allows us to see the impact of memory cells. Upon the first exposure to antigen A, antibody concentration increases to a maximum after 14 days. But upon a second exposure to antigen A, 1,000 times more antibodies are produced within 10 days. Upon a second exposure, memory B cells are responsible for the release of greater quantities of antibody in a shorter period of time. Memory cells provide long-term immunity to the pathogen. Here are 3 IB syllabus statements that should be well within your control. B cells secrete antibodies. B lymphocytes are activated by T lymphocytes in mammals. And activated B cells multiply to form clones of plasma cells and memory cells. And could you explain how immunity depends on the persistence of memory cells? You can use this graph. Memory cells reside in the body after a first exposure to an antigen, whether the exposure be an infection or a vaccine. Keep that in mind. Whether that first exposure be an infection or a vaccine. Memory cells are waiting, ready for a second exposure following a first exposure. When the second exposure occurs, more antibodies are released over a shorter period of time relative to the first exposure. The secondary immune response is so strong that you fight off the infection without even knowing you have encountered the pathogen for the second time. This is the basis of immunity. So let's end this movie with a closer look at antibodies. Explain how antibodies aid the destruction of pathogens. Antibodies are Y-shaped proteins with 4th-level structure, quaternary structure, because they're composed of 2 or more polypeptide chains. B cells make antibodies specific to the antigen, and antibodies serve to neutralize the antigen. The Y-shaped antibody is composed of 4 polypeptide chains, 2 heavy and 2 light chains. The antigen binding site is here. It's a region that's variable among antibodies. The variability of this region is what gives antibodies their specificity to bind certain antigens. And this region of the antibody is the constant region. Antibodies can be secreted into the bloodstream where they circulate freely, or antibodies can be bound to the membrane of B cells. As you can see on the left, and on the right you can see the antibody embedded in the membrane of the B cell. You can see the antigen binding sites here, and each specific antibody will have a different variable region providing the specificity of antibody-antigen binding. In this image, you can see the antibody that is typical of T cells. Not all antibodies are constructed as Y-shaped molecules. You can see the antigen binding site here. The high specificity of our immune system to the numerous pathogens we encounter can be explained through RNA processing and alternative splicing of raw mRNA transcripts. Alternative splicing of mRNA transcripts can result in an extremely large number of unique lymphocytes. We encounter numerous pathogens through the course of our lives, yet we have specific lymphocytes ready to be activated upon the exposure to any pathogen. Antibodies, seen here, will bind to the antigens on the surface of the pathogen, and you can see the molecular specificity of the antibody-antigen relationship. Antibodies, once they bind to the antigens on the surface of the pathogen, will neutralize the activity of pathogens or clump the pathogens together, facilitating phagocytosis by macrophages. By binding to viruses, the antibodies neutralize the virus by preventing the virus from entering a cell. Antibodies bind to pathogens, causing them to clump, and this facilitates phagocytosis by macrophages or other phagocytes. In this diagram, you can see a macrophage engulfing pathogens that have been bound up by antibodies. Hopefully, you can describe how plasma cells, B cells or B lymphocytes, secrete antibodies. Now, as a part of your response to this statement, you can go into detail about protein synthesis, modification of protein by the Golgi, and secretion of the protein by the cell through exocytosis. You need to also go into detail about B cells being activated by helper T cells. You should also mention the formation of a clone of cells, all of which secrete antibodies. You could mention memory cells that linger in the body over long periods of time, ready to respond upon a second exposure to the pathogen. And the second statement is, explain how antibodies aid the destruction of pathogens. Review the last 9 slides to strengthen your control of this statement. You might remember from our study of genetics that molecules on the surface of our red blood cells distinguish us as either type A, B, O, or AB. The molecules on the surface of our red blood cells are potential antigens if blood is donated to a person of a different blood type, and each blood group produces specific antibodies in response to the antigens. Thus, we must be very careful about blood transfusions, because if type B blood is donated to a type A person, type A antibodies will bind the type B cells, causing clumping of blood in the recipient, potentially leading to death. And lastly, let's make a connection to topic 6. HIV is a virus that infects helper T cells. And remember that viruses lack a metabolism. Viruses infect cells and use the cellular machinery of the host cell. Antibiotics block metabolism in prokaryotic cells, but not viruses, because viruses lack their own internal metabolism. So could you outline the effects of HIV on the immune system? Could you state that there's a reduction of active lymphocytes, helper T cells, and a reduction in antibody production, because without helper T cells, B cells are not activated, and this would lead to the development of acquired immune deficiency syndrome. HIV infects helper T cells most significantly, some macrophages and some B lymphocytes. With a decline in helper T cells, there's a real loss of active lymphocytes and a reduction in antibody production. Remember, helper T cells are stimulated directly by macrophages. Helper T cells stimulate both B cells and cytotoxic T cells. HIV reduces the number of helper T cells, and thus the entire immune system is compromised. This is human immunodeficiency virus. This graph displays helper T cell concentration across time following HIV infection. You can see the number of helper T cells declines over time. HIV, by infecting helper T cells, compromises the entire immune system. And that brings us to the end of IB Bio, Topic 11, Part 1, Immune System A.